Welcome to this video, an introduction to pharmacokinetic drug-drug interactions. The intention here is for this video to accompany a series of videos on this channel on pharmacokinetics, and I'm trying to put pharmacokinetics into a wider context. Now, it is important to understand the contents of this current video that you've seen other videos on this channel, the basics of drug metabolism and transporters, genetic polymorphism in drug metabolism, and since we're going to bring together all our knowledge of pharmacokinetics here, then you should be familiar with the videos where we calculated pharmacokinetic parameters. That's videos 1 to 7. So it is important you are familiar with the contents of those videos to understand what's going on currently. Now, when we calculated those pharmacokinetic parameters in the previous videos, they aren't just abstract numbers. They tell us something about what's happening with the drug as it passes through the body. In this video, we will try and put those pharmacokinetics into a clinical setting. And to do that, we're going to take some research data, apply our knowledge of pharmacokinetics to interpret that data, and come to certain conclusions about what's happening with the drug. So I do hope that you will find that particularly interesting. In those previous videos, we calculated the pharmacokinetic parameters, but we assumed that we were dealing with an average or a normal population, whatever normal means. There are various factors that can change the pharmacokinetics of a drug. We've met two previously, age and liver disease. But apart from those, the most important factors are genetics, or genetic polymorphism, which we covered in a separate video, and interactions with other drugs. And it is those so-called drug-drug interactions that we're going to cover here. First of all, let's just clarify what we mean by a drug-drug interaction. It is perhaps better to call them xenobiotic interactions, although that is a little bit of a mouthful. There are certainly interactions between different drugs, but you can also get interactions between drugs and food, drugs and herbs, and drugs and alcohol. I know what you're thinking, food, herbs, alcohol, aren't we kind of splitting hairs here a little bit? Maybe we are. And so we tend to use a more generic term, drug interactions, without actually defining what the drug is interacting with. And I do use that term in this video. Let's look at the principle behind a drug interaction. It occurs when one drug, or to be complete, a drug or a xenobiotic, alters the pharmacology of another drug. Let's take an example. Some person takes drug A, and that drug generates some pharmacokinetic profile. That is a time versus drug concentration curve, such as the one shown here. And in fact, in videos 1 to 7, this is pretty much what we dealt with. We took the time versus drug concentration curves and calculated various pharmacokinetic parameters from that profile. Let's take a slightly different situation where this person takes drug A, but also takes drug B at the same time. Drug B has some effect on the biochemistry or physiology of this person that results in an altered pharmacokinetics of drug A. So that's it as general principle, but there are different types of drug interaction that are represented on this slide. Probably the most common and most important is where you inhibit some enzyme or membrane transporter, and that inhibition is usually competitive inhibition. Now, we'll come to that in a little bit more detail in a moment, But let's first of all pick out some of these other mechanisms here, which are perhaps not as common, but we do need just to cover them for completeness. And we'll start with the displacement of bound drug. An example of that is with warfarin. Warfarin is 99% bound mostly to serum albumin. In this slide, a blue dot represents warfarin. The serum albumin is represented by that sort of brown splodge. And you can see the warfarin bound onto the serum albumin. In actual fact, there is an equilibrium between the bound and unbound warfarin. And the unbound fraction, which is 1% in this particular case, the unbound fraction is designated Fu, and it's the unbound fraction which is responsible for the pharmacology of warfarin, and it's also the fraction which is cleared. The pharmacology of warfarin, of course, concerns an inhibition of blood clotting, so warfarin is sometimes called a blood thinner for that reason. Fraction unbound was covered in video 6 part B on clearance, and we use that Fu factor there in a number of equations in that video. Now let's consider a slightly different situation where another drug is taken along with the warfarin. And the other drug in this case is clofibrate, which is a lipid-lowering drug. What happens here is that clofibrate binds onto the protein and displaces some of the warfarin. So now the unbound fraction of warfarin has increased. And since it's the unbound fraction which is responsible for the pharmacology, that then results in an increase in blood clotting time. With warfarin, that can be important because warfarin has a very narrow therapeutic window, and so the fraction unbound might exceed the minimum toxic concentration. And if you're not sure about therapeutic window, it was covered in video 7 on repeat dosing. Another type of drug interaction is induction, which is the sort of opposite of inhibition. Induction occurs post-DNA translation and can result from, for example, an increase in transcription of messenger RNA. An example is St. John's wort. That shrub, the picture of that shrub there is St. John's wort that used to grow in my garden. Just thought I'd mention it. St. John's wort has been used as a herbal antidepressant, and it's been called nature's Prozac, which sounds great, except it can cause some pretty severe interactions with medications. The active ingredient in St. John's wort is hyperforin, and it's a potent upregulator of CYP3A4. If you're not sure what CYP3A4 is, it is covered in the video on the drug metabolizing enzymes and transporters. So, what are the consequences of upregulating CYP3A4? That enzyme is implicated in the metabolism of 50 to 60% of drugs on the market. If you increase the CYP3A4 activity, that increases drug clearance, that lowers the plasma concentration, and therefore lowers the therapeutic effect of the drug. There are some very important classes of drug which are metabolized by CYP3A4. They include oral contraceptives, cyclosporin, which is an immunosuppressant very often used after organ transplant, and a number of antivirals. Now, if you lower the therapeutic effect of an oral contraceptive, then the result can be fairly obvious. But if you lower the therapeutic effect of some of these other drugs, then the result can be catastrophic. Going back to the types of drug interaction, there is, along with competitive inhibition, there is also non-reversible inhibition. Again, it's not very common, but I think we'll come back to that in a minute. And what we'll deal with first is competitive inhibition. Now, this diagram you've seen before, video 6 part A on clearance, where a drug represented by the blue spots here is carried in the bloodstream to the liver being the main organ of metabolism. And so in the liver you have a series of enzymes, and our drug binds onto the active site of the enzyme where there is a reaction, and the metabolite thus formed is then cleared. And so I've just described hepatic clearance by metabolism, as described in video 6 part A. Now let's consider a slightly different situation again, this time where you have another drug in the bloodstream. So we have our blue dot drug, but we also have this pink dot drug as well. And our pink drug now binds onto the enzyme and it blocks the blue dot drug. Now, it doesn't block it entirely, it competes for the active site. And that is classic competitive enzyme inhibition. So you still get the metabolite formed, and the metabolite is still cleared, but it's much slower than it was before. So you have now slowed down the clearance of the metabolite from the bloodstream. So that's competitive inhibition. Now we can return to our classification and look at a little bit more detail on non-reversible inhibition. Irreversible or non-competitive inhibition, various names of the same thing, is relatively unusual. But instead of a drug competing for the binding site, the drug binds very strongly. It forms a tightly bound complex, or even occasionally it covalently binds into the binding site. An example, macrolide antibiotics such as erythromycin, clarithromycin, antibiotics of that type, they can irreversibly bind to the CYP3A class enzymes. In fact, the irreversible inhibition of CYP3A4 by clarithromycin, which then affects Midazolam clearance, has been reported. In such cases, the activity of the enzyme only returns with protein turnover. So the inactive enzyme is broken down and removed and replaced by newly synthesized enzyme over time. Now, don't get the idea that drug interactions are only with CYP enzymes. Far from it. It can occur with virtually any transporter enzyme, and I've just given an absolute tiny snapshot here of examples. So you have a CYP enzyme, you've got a transporter protein, and you've got a phase 2 enzyme involved in glucuronidation there as well. So I'm not going to go through that in any detail. I've just given examples to show that these drug interactions can potentially occur with any protein, in actual fact. Now, as promised, let's turn to a clinical research study. And this study looked at some drug-drug interactions. These were drug-drug, not drug-herb or anything else. They were two drugs interacting. The study compared the pharmacokinetics of 4 drugs before and after the co-administration of other drugs. And we will see how we can interpret the pharmacokinetics that we obtain from this study to tell us something about what's going on. The study was done by a PhD student of mine, and I should point out to start with that the study, which it was reported, there's the reference there, you can get the paper and have a look. But the study wasn't specifically about those drug-drug interactions. It was more of a proof of concept study for a method of studying drug-drug interactions. So it's a methods paper rather than specifically on drug-drug interactions. But the data are in there, and you can see them, and the data I'm about to present are in that publication. Now, drug-drug interactions, they are often looked at in terms of the victim and perpetrator drugs. The victim drugs are those having the altered pharmacokinetics, and the perpetrator drugs are those causing the changes. The perpetrator drugs in this particular study, they were inhibitors, and it was ketoconazole and fluvoxamine that was used. The victim drugs in this study were midazolam, tolbutamide, caffeine, and flexophenidine. Now, I'm not overly concerned about the action of these perpetrator drugs themselves. I mentioned ketoconazole and fluvoxamine in the video which is about to come, but overall we just consider these as perpetrator drugs. Our focus is on the pharmacokinetics, or more importantly the changing pharmacokinetics of those 4 victim drugs. What was the experimental outline? It was a 2-stage study. In stage 1, 6 healthy male volunteers were given single doses of midazolam, tolbutamide, caffeine, and flexophenidine. These were by themselves without any perpetrator drugs. The purpose was to obtain baseline plasma drug concentration versus time pharmacokinetic profiles. We wanted to look at the pharmacokinetics of those drugs by themselves. Now, the pharmacokinetics of those drugs is fairly well known. There's plenty of papers in the literature, and so we wanted to make sure that the pharmacokinetics that we observed fitted into the general literature values. And so we did do that comparison. Those literature values will be given in the course of this video, but I don't focus on them. They're just there for comparison. Now, after stage 1, there was what's known in the jargon of these types of studies a washout period. It's just a period of time which is long enough to allow those drugs to be removed, to be eliminated from the body, to a point where you can't detect them any further. And once we got to that point, we went to stage 2, where we gave the single oral doses of those 4 drugs again, but this time they were co-administered with the perpetrators. Those perpetrators were actually given as repeat doses to maximise the effect, but that is somewhat beside the point. They were given to the same 6 healthy male volunteers, and then we looked at the pharmacokinetics in stage 2 to see how it had been affected by those perpetrator drugs. So let's have a look at each of those victim drugs one by one. First of all, midazolam. There's midazolam alone, and there's midazolam with the perpetrators. And you can see immediately that there is a big change in the pharmacokinetics. Let's look at the pharmacokinetic parameters to quantify those changes. Now remember, these were administered orally. There was no intravenous dose, so we were unable on this study to calculate clearance and volume of distribution. Clearance and volume of distribution is known for these 4 drugs, And so those values are available. It's just that we couldn't do it on this particular study. So you can look at the Tmax, you can look at the half-life, you can look at Cmax, and you can look at the area under the curve. So first of all, Tmax, no change. The Tmax wasn't affected by the perpetrators. But the half-life was very significantly increased. There was a 4.5-fold increase in half-life caused by the perpetrators. Cmax and AUC, out of those two parameters, the area under the curve, the AUC, is probably the important one, because what does AUC mean? What does it signify? Altogether now, exposure. So in this case, those perpetrators caused a nearly 13-fold increase in drug exposure. Midazolam, of course, is a sedative. It's used as a short-term sedative in procedures such as dentistry or colonoscopy. And you can see from Midazolam alone that it's cleared fairly quickly, and so the effects wear off. Now you can imagine if you went in for some dentistry and you took some Midazolam, but you had these perpetrator drugs circulating in your system, you are going to be sedated for a very much longer time. So what's going on with Midazolam? Well, Midazolam is cleared through metabolism, and Midazolam is cleared by a reaction with CYP3A4. It's a hydroxylation. There are then subsequent metabolic reactions before it's cleared, but it's initiated by CYP3A4. And it is CYP3A4 that is being inhibited, and it's being inhibited by ketoconazole, which is a strong CYP3A4 inhibitor, and so that results in reduced plasma clearance. We look at that little sort of cartoon that I used to explain competitive enzyme inhibition. You can see the pink dot blocking the yellow dot on the enzyme. If you now look at that in respect to CYP3A4, you can see the iron-containing heme group there, and a slightly greyed out structure is the ketoconazole bound into the active site. So, you know, that cartoon of enzyme competitive inhibition wasn't really far from what is really happening. And I would just remind you about the video on the basics of drug metabolism and membrane transporters, which explains all this in a little bit more depth. Let's go on to the next compound. My morning coffee in my nerdy mug, and we're moving on to caffeine. This is caffeine alone. This is caffeine with the perpetrators, and again you can see a very significant change in the pharmacokinetics. Let's look at the pharmacokinetic parameters. Cmax is not changed. Half-life, there's a 6.3-fold increase in the half-life of caffeine. And if we look particularly at the AUC there, that results in a just over 8-fold increase in exposure. Caffeine, of course, is a stimulant, and if you take caffeine along with those perpetrators, you are increasing the effects of that stimulant quite dramatically. What is going on with caffeine? The major metabolic pathway for caffeine is CYP1A2, which leads to an N-demethylated metabolite, and that metabolite then undergoes other metabolism but is ultimately cleared from plasma. And the CYP1A2 is being inhibited by fluvoxamine, which is a strong CYP1A2 inhibitor, and so that results in reduced plasma clearance. Let's just compare midazolam and caffeine. This is an interesting bit of pharmacokinetic interpretation because you can see they both have an increase in half-life and the area under the curve because the perpetrators have reduced the clearance of those drugs. But there is also a difference here in their Cmax. Why is the Cmax dropped for midazolam and not caffeine? Well, why don't you pause the video and have a little think about that and see if you can come up with an answer. I will just carry on going and tell you really what's happening here. And it goes all the way back to video 1, which was on the foundations of pharmacokinetics. The plasma drug concentration at any one time is an equilibrium between the rate of absorption and the rate of elimination. And so during the absorption phase, the drug is still being eliminated. As soon as the drug gets into the plasma, elimination begins. So absorption, behind absorption, there's a little bit of elimination going on. And of course, behind the elimination, there's also a little bit of absorption going along. And the shape of that curve, of that pharmacokinetic curve, is due to the equilibrium in those various stages. Now, the clearance of midazolam is about 5 times more than caffeine. And so relatively more midazolam is being eliminated in the absorption phase compared to caffeine. And so because a significant amount of midazolam is eliminated during the absorption phase, that is reflected in the Cmax, but relatively little caffeine is eliminated in the absorption phase. Some of it is, but relatively little compared with midazolam, and so that is also reflected in the Cmax. And when you inhibit the clearance, which is a relatively low number anyway, it doesn't have that much effect on the Cmax compared what it does with midazolam. So you get that change because of the greater value of clearance for midazolam. Let's move on to the next drug, which was Tolbutamide. That is Tolbutamide alone. That's Tolbutamide with the perpetrators. Now you can see there's an effect, but the magnitude of that effect is not as great as it was with the other drugs. Let's look at the pharmacokinetic parameters. Again, no change in Tmax. A small change in half-life, about doubled. And maybe exposure is about doubled as well. So there is an effect, but that effect is not as great as it is with the other drugs. What's going on with Tolbutamide? It's cleared through CYP2C9 to the hydroxy metabolite, which is then cleared from plasma. And so we're looking at the inhibition of CYP2C9. That occurs with fluvoxamine, but it's only a moderate inhibitor. And so consequently, you only get a mildly reduced plasma clearance. So in this case, there's a quantitative difference in the change of the pharmacokinetics compared with the other drugs. Let's just go back to those pharmacokinetic parameters, and the really observant amongst you may notice something here, and that is n equals 4. This is only 4 subjects. Now, I did say that we administered 6 subjects, and indeed we did give 6 subjects Tolbutamide. These subjects were all genotyped. And when we looked at the CYP2C9 genotypes, we discovered 2 subjects were poor metabolizers. 4 were wild type, 2 were poor metabolizers. Now, if you're not sure what poor metabolizers mean, you need to look at the video on polymorphism. Now, the pharmacokinetic profiles was different between the wild types and the poor metabolizers. So if I put the whole thing up, you can see it here. The red and the blue lines are the same data you saw previously for the wild type. The green are the poor metabolizers, N equals 2 in this case, and that was Tolbutamide alone. So there is a significant effect of these subjects being poor metabolizers. But then when you give the poor metabolizers the perpetrator drugs, then you're seeing a really major change in the pharmacokinetics. So what you're seeing here is an example of a drug-drug interaction operating alongside a genetic polymorphism. So that just goes to show the sort of complications you get in populations where they may be taking different drugs, or indeed taken in different xenobiotics based on their diet, and of course they will have different genetics. Moving on to the final drug, which is Flexophenidine. So that's Flexophenidine alone. That's Flexophenidine with the perpetrators. This is the pharmacokinetics. Again, no real change in Tmax. Half-life, well, I'm saying there's no significant change. It's actually very hard to come up with a single number for half-life because the elimination is polyexponential. If you look at the literature, half-lives anywhere between 3 and 17 hours have been reported, depending upon how they've done the calculation. So what we're doing here is we are looking at the shape of the curve, and by the shape you can see Flexophenidine alone and that co-administered with the perpetrators, they're parallel lines, they're parallel curves, so there's no change in the half-life. There is a difference in exposure of just over 3-fold. So what's going on here with Flexophenidine? It's clearly somewhat different to the other drugs. What's happening? Well, Flexophenidine is not metabolized. It's excreted in urine as unchanged compound. If you look at the video where we looked at drug metabolism and transporters, you were introduced to P-glycoprotein, or PGP, which is an efflux transporter. So what that means is that Flexophenidine, which is the structure shown there, goes through the membrane into the cell, and then the PGP comes along and pumps Flexophenidine out of the cell. It is an efflux transporter. So Flexophenidine is being pumped out of the cell by PGP. Ketoconazole, in this case, inhibits the PGP, and therefore less Flexophenidine is pumped out, so the overall absorption increases. If you compare Midazolam with Flexophenidine, both have an increase in Cmax, and both of them have an increase in the area under the curve, or an increase in exposure. But the reason for those increases are different for the two drugs. Midazolam has an increase in half-life because it's the enzyme of elimination that it is inhibited. But that's not the case for Flexophenidine, which has no increase in half-life, because what's being inhibited here is the efflux transporter, which is involved in absorption. Now, we've looked at drug-drug interactions. We looked at a drug-herb interaction, which was St. John's wort. Let's just have a look at an everyday food-drug interaction. And in fact, all foods contain xenobiotics of some type or another. And where there are xenobiotics, there is an opportunity for interactions with medication. And I want to just use one of them, one example, which is grapefruit. Grapefruit contains a series of compounds called furan coumarins, which are inhibitors of CYP3A4. We're back to CYP3A4 again, which is featured a lot in this video, but it also inhibits certain other enzymes as well. The effects of grapefruit can vary greatly upon the brand, the source of the grapefruit, its preparation, etc. But its effect was discovered by accident. Grapefruit juice was used to mask the taste of alcohol in a drug-alcohol interaction study. And when they put the grapefruit in, they found significant effects on the pharmacokinetics. In actual fact, it is an example of irreversible drug interaction, and the inhibition effects of grapefruit only disappear with protein turnover. Now, to me, this is one of the great ironies, because you go online and you can look at detox diets, and you will find grapefruit detox diets, which, in my opinion, are complete rubbish, and in fact do the exact opposite, because what the grapefruit juice is doing in those diets is inhibiting the enzymes, CYP3A4 in particular, which are involved in the natural clearance of potentially toxic xenobiotics. So a grapefruit detox diet is doing the exact opposite of what's being claimed. Let's move on quickly and point out there are many drugs where patients are advised to avoid grapefruit. These are the ones off the UK NHS website. I'm not going through them. You can look them up for yourself, but there's a long, long list. When you're doing research studies, clinical trials, and indeed the pharmacokinetics study that we looked at on this video, the protocol will say that the volunteers should not be having a grapefruit because it does interfere with the pharmacokinetics. That just leaves us to sum up. There are various types of drug interaction. We looked at displacement of bound drug, and the example we looked at was warfarin with clofibrate. We looked at induction, and the example we looked at there was St. John's wort. We looked at non-reversible inhibition, which was our example, in that case, was grapefruit. And our focus here was on competitive inhibition, and we looked at a research study. And we looked at the effects of ketoconazole and fluvoxamine inhibiting Midazolam, Caffeine, Tolbutamide and Flexophenidine. We had an increase in half-life and area under the curve for Midazolam, Caffeine and Tolbutamide, which was caused by the competitive inhibition of certain CYP enzymes, which decreased their clearance. And we saw an increase in area under the curve for Flexophenidine, but not the half-life. And that was caused by competitive inhibition of the PGP efflux transporter. In other words, it was an effect on absorption. So that's the end of this video. I do hope that you enjoyed it, and I wish you farewell.